Okay, let's continue with the senses, dealing with hearing and equilibrium. Uh, Now, these are pretty similar um, senses because they both work with what are called hair cells with stereocilia and their mechanoreceptors. So first of all, talk about the nature of sound. What is sound? Compression of airwaves and rarefaction of airwaves. So it comes at you in waves of compressed air molecules and rarefied air molecules. And the pitch, how high it is or how low it is due to the frequency. How many waves per second pass a certain point. So that's pitch. And we'll talk about how the ear and brain determine pitch. And then loudness is in decibels. That's how big the wave is. Right? Okay, ear anatomy. So they got three parts of the ear anatomy, which we learn in lab: the outer ear, the middle ear, and the inner ear. The outer ear is the pinna and external auditory meatus until we get to the tympanic membrane. On the other side of the tympanic membrane is the middle ear. And attached to the tympanic membrane are three middle ear ossicles, the smallest bones in the body, the malleus, the incus, and the stapes. And then there's a eustachian tube that leads to the nasopharynx. And there's a couple of muscles in there called the tympani and the stapedius that will harden the ossicles if you hear a really loud noise with a millisecond or so delay. Two of the middle ear muscles have a really interesting evolutionary history. They're actually evolved from reptilian jawbones. Ancient reptiles had three bones in their mandible, in the lower jaw. We have one. Mammals have one. Those two bones that were back here in the mandible moved up into the middle ear and became two out of the three middle ear ossicles. Kind of strange, but that's what. But they're actually quite a good evolutionary record of these bones being later and later reptiles being smaller and smaller and smaller, and finally ending up in the ear in mammals. Okay. The inner ear. The bony and membranous labyrinth. The bony labyrinth is this, we know what a labyrinth is a complicated series of passageways, and that's the temporal bone. Because your entire inner ear, and middle ear for that fact, is encased in the temporal bone. And you hear waves through your external auditory meatus that vibrate your tympanic membrane, your eardrum. But you also feel vibrations of the temporal bone actually will be transmitted to your inner ear. The membranous labyrinth is what's called the vestibule, the semicircular canals, and the cochlea. The vestibule and semicircular canals deal with the equilibrium. The cochlea deals with hearing, and they are complicated passageways inside there, also filled with fluid. And it's lined by a membrane, so it's called the membranous labyrinth. Now, in the cochlea, there's what is three ducts, but there are really only two. The scala tympani and the scala vestibuli are the same one, really. They start on one side of the cochlea, they go up the cochlea, and they hit something called the helicotrema, and then they come back down. It's the same fluid in both. They're continuous. But they look like different. When you cut the cochlea in cross section, it looks like two different chambers. Chamber in the middle is called the scala media, or the cochlear duct. That is filled with a strange fluid called endolymph, and I'll talk about why it's strange in a moment. And the scala tympani and vestibule are filled with something called perilymph. Now, in the cochlear duct is something called the spiral organ or organ of Corti. And this is where the hair cells and supporting cells, the actual auditory receptors are located. They are on this membrane called the basilar membrane that separates the cochlear duct from the vestibule. And the base of the hair cells is on the basilar membrane. And these hair cells are hair cells because they have these stereocilia, these stiff hairs that point out from them. And these hair cells are stuck in another membrane called the tectorial membrane. And we also have to talk about the vestibular membrane. I'm going to show you a cross section later on inside, and you'll need to know the orientation of these membranes. So there is a cross section of the, so here's the auricle or pinna, external auditory meatus, tympanic membrane, and here's the middle ear right here. The three middle ear ossicles, the malleus, sometimes called the hammer because it's shaped like a hammer, the incus, sometimes called the anvil because it's shaped like an anvil, and the stirrup, the stapes, sometimes called the stirrups because it's shaped like stirrups. And the so the malleus is attached to the tympanic membrane. The stapes is attached to what's called the oval membrane. The oval membrane is much smaller than the tympanic membrane. So in essence, the sound waves are amplified from the tympanic membrane to the oval membrane by the middle ear ossicles because the oval membrane is much smaller. So you have this larger membrane that getting pushed back and forth, and then you're pushing, and that will be amplified to a greater push back and forth on the oval window. You have to amplify the sound waves because you got to translate compression and rarefaction of air molecules to compression and rarefaction of paralymph to a liquid in the scala tympani, so the stipuli, that is paralymph, is liquid. So you have to convert. So it's much harder to push fluid than it is to push air. So you have to amplify it. Okay, then it enters the cochlea, goes all the way up this spiral. The waves go all the way up this spiral to the top to a place called the helicotrema. They come back down, and they leave via another window called a round window. So the waves enter the cochlea at the oval window. They leave the cochlea at the round window. Okay. And here is the vestibule right here, and this we're going to get to that in equilibrium, and the semicircular canals will get to those at equilibrium also. And then there's the vestibular cochlear nerve. Right here, it's cranial nerve 8. There's a vestibular branch and a cochlear branch, and it's collectively called the vestibular cochlear nerve that carries equilibrium information and hearing information into your brain. They don't show the two little muscles in here. One of them is attached to the malleus, called the stapedius. One of them is attached to the stapes, I think, and is down in the auditory tube here, called the tensor tympani. The auditory tube is also known as the eustachian tube. Both of those are correct names for that tube. Okay. So physiology of your function middle ear ossicles, as I mentioned, to amplify the sound waves. All right. Tympanic reflex. This is when those two little muscles come into play. If you hear a really, really loud sound, these two that will go into a nucleus, I think, in your medulla oblongata, and immediately come motor neurons will immediately come out and contract these two small muscles to stabilize the ossicles so they don't move too much. Because if they move too much and cause too violent movement of the paralymph inside the cochlea, you will break the stereocilia on your hair cells. And we all have broken stereocilia. We all have damage due to the loud noises. Because ears did not evolve to handle the really loud noises of industrial society. You know, what's the loudest thing in nature? Maybe a thunderclap or something. So we all, depending on how many rock concerts you've been to, or how many jackhammers you listen to, or how many, or gunshots. If you shoot a gun, you should always wear ear protection. And I like to go to. I might wear some ear protection if I go to my next NFL football game, because it's just really loud, and they really encourage people to be as loud as possible. Every decibel counts, as they say in M&T Stadium. Every decibel counts, and it's over 100 dBs. So this is causing ear damage. I was really this is when Aaron Rodgers won the Super Bowl, and he's there, and so he's holding his little infant son with him in the stadium, and he's got earphones on his son, because it's 100 dBs in the stadium. And this is a little tiny infant, and it's going to cause ear damage for the rest of his life. I was at, believe it or not, I was at a tractor pull. You can believe I was at a tractor pull. It was so loud because these engines have no mufflers on. And they had these parents with these little children in the stands with incredible noise. I mean, you're just causing ear damage on these kids for the rest of their life. There's a bunch of damage. These are nerve cells. You damage them, they don't get repaired. Okay, and everybody's got, and there's a condition called tinnitus, where you have this ringing, this loud ringing in your ears, and it's there all the time because of the damage to hair cells. And we all have a little bit of that. Some people have more than others, depending upon how much. And there have been rock stars who've complained in their later life about having incredible hearing loss because they played their music at, a couple hundred dBs for years. Okay. Stimulation of cochlear hair cells. Vibration of paralymph in the scala vestibuli to the vestibular membrane, which causes fluid movement of endolymph in the cochlear duct, which vibrates the basilar membrane. Right. The stereocilia in the hair cells are embedded in the tectorial membrane that doesn't move. So you have this tectorial membrane that doesn't move, and here you have the basilar membrane with the base of the hair cells, and it's moving up and down. And the stereocilia are stuck in this membrane. So as the basilar membrane moves up and down, the stereocilia are bent like this. And at the base are potassium ion channels that open up and cause potassium to rush into the cell and depolarize the cell and cause a receptor potential. So hair cells are mechanoreceptors. The deformation of the membrane of the hair cell is going to open up ion channels, and that's going to cause a receptor potential. And that will lead to an action potential and a primary sensory neuron that goes into the vestibular cochlear nerve into your brain. So bending of stereocilia causes opening of potassium channels that leads to depolarization of hair cells that leads to APs in the sensory neuron. Okay, that statement should give you pause. All right. Here, let me jump to this one first. Down here. What's wrong with this picture here? From chapter 12, the nervous system, what do we know about what happens when we open up potassium ion channels in nerve cells? What direction does the potassium go? Out. It's the repolarization phase of the action potential. So potassium, and why is potassium leave the cell? Because it's more concentrated inside the cell than outside. And it leads to a hyperpolarization, a repolarization or a hyperpolarization. Well, is this a mistake? Is this an error? This has potassium going into the hair cells and causing a depolarization. And this is why, because of the bizarre nature of endolymph. Believe it or not, endolymph has a very high potassium concentration, more so than the hair cell, than the potassium concentration inside the hair cell. It's a truly bizarre liquid. So the concentration gradient for potassium is into the cells. And of course, potassium is a positively charged ion, so it depolarizes the cell. Truly bizarre. All right, goes against everything we learned about it in the nervous system. But that's the way that hair cells work. All right. Let's go back and look at this cross section of the cochlea. See, here's the cochlea right here, and we're taking a cross section of this. And remember, it's a spiral thing. And so here's a scala vestibuli, which is in contact with the oval window. Here's the vestibular membrane here. And this blue stuff is perilymph. Here's the endolymph in the cochlear duct. Here's the tectorial membrane right here, and the basal membrane down here. You blow that up, and here it is. Tectorial membrane here. Here's the hair stereocilia that are embedded in it. And remember, this membrane doesn't move. There's some supporting cells that are helping the hair cells. And here's the hair cells. There's an inner set of hair cells and an outer set. We don't have to worry about that. And here's actually an electron micrograph of it. And so this movement of the perilymph and invasive scala vestibuli moves the vestibular membrane, which moves the endolymph in the cochlear duct, which causes the basal membrane to go up and down, which causes the stereocilia to be bent. Every time the basal membrane goes up, it bends the hairostilia. Potassium ions opened up. If the stereocilia bent, potassium ushers into the cell, depolarizes, and leads to a receptive potential. And there it is, the stereocilia embedded in the tectorial membrane, endolymph up here. Here's the basal membrane down here. And these tip links mean the stereocilia are attached. And once you bend the stereocilia, as I said, potassium channels opened up, potassium enters the cell, depolarizes. Okay. Oh, and that leads to a receptor potential that stimulates these dendrites to threshold, and you generate an action potential on the cochlear branch of the vestibular cochlear nerve. Okay. So here's amplitude in the external ear. Here's the waves in the middle ear amplified due to the middle ear ossicles. So here you can see that the oval window is here, and the waves come in here, right? Sound waves enter ear. Tympanic membrane vibration moves auditory ossicles. Sound waves are amplified. They're sent into the scala vestibuli across the oval window, and they move down the scala vestibuli, right? And it just so happens that so pressure waves cause the vestibular membrane to move, resulting in pressure wave formation in endolymph within the cochlear duct, displacement of a specific region of the basal membrane. Here's the thing: the basal membrane is frequency coded. Each frequency we only move a particular part of the basal membrane, and that's how you detect what pitch is. Right? So these waves will come in here, and they're causing waves all along here in the cochlear duct, but only this part of the basal membrane is sensitive to that frequency. Right? So only this part of the basal membrane vibrates. Right? Now this is a little misleading, because these waves are going to continue in the scala vestibuli all the way up here around what's called the helicotrema. And they're going to continue down, continue down here in the scala tympani and all the way out right around window. It shows only these waves leaving. Well, these waves are going to go all the way around here, but they're not going to cause any more stimulation, because that part of the basal membrane is not sensitive to that frequency. So this picture is a little misleading. Okay, and then the waves, remaining pressure waves are transferred to the scala tympani and exit the inner ear via the round window. Okay. So sensory coding, loudness, amount stereocilia are bent results in changes in frequency of APs in the primary sensory neuron. So the bigger the waves, the more the basal membrane vibrates, the greater the stereocilia are bent, the more ions enter the membrane, the higher the receptor potential, the higher the frequency of action potentials along the primary sensory neuron. So that's how your brain knows whether it's a soft sound or a loud sound. Right? Pitch, basal membrane frequency coded. High frequencies near proximal end, low sound frequencies near distal end. And so I'm going to show you that in a moment. So as I said, the basal membrane is frequency coded. All different, a particular frequency will only stimulate or will only cause part of the basal membrane to vibrate. And that's how your brain knows what the pitch is. Sound focusing. Now, pons can send efferent information to inner ear to dampen vibrations of basal memory to focus in on something. Here's how you can zero in on some sounds in a mixture of sounds. By the sound focusing, it's a little bit of efferent information. Which means from the brain to the basal membrane, and you can actually dampen certain areas of the basal membrane to a certain degree. You can't dampen it entirely. So say you come into a room full of people talking, and all of a sudden you hear some voice you recognize, or somebody who's saying something particularly interesting. You can kind of focus in on that one sound, and listen to it a little more intently than the rest of the sounds around you. That's sound focusing. That's dampened vibrations of the basal membrane for the other frequencies, except for the frequencies of the person you want to hear. Okay. So there's the basal membrane right there. High frequency sounds are coded around the base of the basal membrane. And that would be about 20,000, I think. Now, I mean, we have a range of what frequencies we can hear. I think we're from about 100 hertz or hertz is, oh yeah, well, there's at the bottom right there, 20,000 hertz, which is cycles per second. That's our limit, a high pitch, all the way down to 20 hertz, 20 per second. And that's that is coded by the very distal end of the basal membrane. Now remember, other animals, particularly like dogs, have a much longer basal membrane, have a much longer frequency range. They can hear things in frequencies much higher than we can. Hence those dog whistles that they can hear, but we can't. They also have a better sense of smell than we have. But I think our eyesight's better than theirs. Okay. Okay, directionary sound. How do you know where sound's coming from? And it's which side of head gets the wave first. So this is pretty impressive, because how fast is sound at sea level? It's about 750 miles per hour, I think. And so you hear a sound over here, and the sound hits this ear first, causes the basal membrane in this ear to start vibrating before it hits this ear. And how distance is that? What is that? That's it's about 10, 15 centimeters or something. You know, it's this far. How fast does it take a sound wave to travel from here to here at going at 750 miles per hour? Well, some fraction of a millisecond, right? But it's still close enough. You can detect it. You know a sound's coming over here, or a sound's coming over here. But what if the sound is directly ahead of you, directly above you, or directly behind you? So you can close your eyes and you can make someone make a sound directly in front of you, above you, or directly behind you, and you won't be able to tell whether it's in front of you, up above you, or behind you, because it hits both ears at the same time. So that's why if you're not sure where a sound is coming from, it's best to turn your head a little bit. And now the sound's hitting one ear before the other one, and you know it's coming from over here. or back there, or up there. Okay, projection pathways. Cochlear nerve from the spiral ganglion becomes part of cranial nerve 8, the vestibular cochlear nerve. To cochlear nucleus of pons to superior olivary nucleus of pons, where efferent information leaves the middle ear muscles. Here's where you have that tympanic reflex that can stiffen the ossicles if you hear a really loud noise. Although some really loud modern noises are too loud and too fast, it doesn't really work, and you end up with ear damage. To inferior colliculus of midbrain to auditory reflexes. Remember the inferior colliculus, it's sound reflexes. All of a sudden there's a loud sound to my right, and I flinch automatically, reflexive, because that information has gone to the inferior colliculus, and I reflexively move to it without even thinking about it. To medial geniculate nucleus of thalamus onto primary auditory cortex and temporal lobe. So here it is. It goes through the thalamus, just like all other senses, except for olfaction. The one other unique thing about the auditory projection pathway is that it has 4 neurons. Every other one has 3. Auditory has 4. Let me skip equilibrium for a moment to show you that. So here we go. Moving the basal membrane produces nerve signals propagated upon cochlear nerve axons, determined in the cochlear nucleus and medulla oblongata. 2 sensory pathways extend from the nucleus. So here's the primary, first sensory neuron here, the primary neuron. It goes to this cochlear nucleus in the pons, and then one branch goes to the superior olivary nucleus. And then they both go to the inferior colliculus right here, remember for auditory reflexes. They both go to the thalamus. So action from septenary neurons in the cochlear nucleus project to the superior olivary nucleus before synapsing all the nucleons. Other neurons are projected to the inferior colliculus. Axons from neurons in the inferior colliculus project to the medial geniculate nucleus of the thalamus. Then axons from the thalamus go to the primary auditory cortex in the temporal lobe, and then they go to primary association with association projections. So look, how many neurons are projected? There's a primary in this pathway, a primary, a secondary, a tertiary, and a quaternary. There are 4 neurons. In this other pathway, there's 1, 2, 3, 4, 5. So in one of these auditory pathways, there's 5 sensory neurons, and one is 4. So that's really different about the auditory projection, because all other sensory pathways have 3, a primary, a secondary, and tertiary. This has a quaternary or possibly a quaternary, or what do you call a 5th one? Well, anyway, a 5th neuron. Okay, let's go back. Okay, equilibrium, static and dynamic equilibrium. Static equilibrium is this. I'm able to stand here, erect, and be stable, all right? Dynamic equilibrium is movement. Which way my head is moving, like this, back and forth. That's dynamic equilibrium. Okay. The organs for static equilibrium are called the sacral and utricle, and they're in the vestibule. And they have hair cells and supporting cells also. They have their hair cells are called the kinocilium and what's called the otolithic membrane. These kinocilia are stuck in a gelatinous membrane that has little rocks in it called otoliths, little calcium concretions, and they weigh it down. So when you lean to one way, this membrane bends to one way, and because the little rocks pull it one way, it bends to the kinocilium, and then you know that I'm leaning to the right here, or I'm leaning backwards, or I'm leaning to the left. And so I can compensate for that and stay upright. So here is the utricle and saccule in the vestibule. Here's the vestibular part of cranial nerve 8. And here are the hair cells. Here's this otoliths in the otolithic membrane. There's the macula. There's the stereocilia and the kinocilium. And see, here's those little otoliths. So remember, you lean a little way and they cause a bending of the stereocilia, open up ion channels. I think it's potassium. Again, this is endolymph. High potassium goes in, receptor potential, and an action potential generated along the vestibular branch of cranial nerve 8. Okay, dynamic equilibrium. This is the semicircular canals. And there are 3 of them. And why are there 3? Well, what kind of a world do we live in here? We live in a 3-dimensional world. We got 3 dimensions. So we got 3 semicircular canals to determine what direction our head, which dimension our head is moving in. Its organ is called the crista ampullaris, which is at the base of each semicircular canal. It's covered by this gelatinous thing called a cupula, and it has hair cells with a kinocilium as well. Its projection pathway, vestibular nerve is part of cranial nerve 8, vestibular nuclei in pons and medulla oblongata, and you send information to nuclei in 5 targets. 1 cerebellum for proprioception, for idea of where your head is in space. Eye muscles. So if you lean in one way, you can compensate with your eye muscles. The reticular formation, the spinal cord, and the thalamus. Spinal cord is to make sure those muscles that are contracting to keep you upright. And it goes again to the thalamus. It's like every other sense except for olfaction and 1 pain pathway. So there is the crista ampulla at the base of each semicircular canal. There it is right there, and there is the cupula, which is a jelly-like. And when you move your head like this, the endolymph, so I move my head like this, and the endolymph is going that way, and the cupula goes the other way. And it bends the cupula. And this bends the hair cells, opens up ion channels, same deal as before. Right? So that is, like I said, head still, head rotating. In what direction is it rotating? And there is the equilibrium projection pathway. And it's really complicated. Look at, as I said before, how many places this thing is going. Equilibrium stimuli are transmitted as nerve signals along the vestibular branch of cochlear nerve 8. Vestibular branch axons projected the vestibular nuclei and the cerebellum. So you know where your head is in space. Equilibrium information is projected through the vestibular spinal tracts to maintain muscle tone and balance. So it goes down a spinal cord. You contract those muscles in your legs, in your back, to hold you upright, to maintain balance. Vestibular nuclei transmit nerve signals to several cranial nerve signals to control reflexive eye and ear movements. So if you move one way, you can direct your eyes in the proper place. Nerve signals are also transmitted to the thalamus and then the cerebral cortex for further processing and for conscious awareness of body position. Right? So they go all 3 of those places. But again, they have 3 neurons. There's 1, there's 2. So these nuclei here, or down the spinal cord, 3 to the thalamus, 2 to the thalamus, and 3 to the cerebral cortex. Okay. So eye movement, nuclei for eye movement, motor coordination, awareness of body position, thalamus, and the vestibular nuclei. Okay. Okay, now we're going to go into vision. So I'm going to take a break here. So this will be, what will this be? 203 senses 3. And then finally we'll hit the eye. Oh, by the way, that's right. In the book, they do eyes before ears. in this PowerPoint, I did ears before eyes, but it shouldn't make any difference. So we'll go in the next video. We'll go on and do visual receptors in the eye, and then that'll be it for this chapter. Okay, I'll catch you in the next lecture. Let me stop recording.